Hello and welcome. In this presentation, we're going to talk about CRISPR and other forms of genetic modification of plants in agriculture. So although CRISPR is now the most common form of genetic modification, it's the most efficient and most widely used today. There are other forms in which humans have been genetically modifying plants throughout history. Some of them are listed here in this image. Some examples are crossbreeding, mutation breeding, transgenic breeding, and then of course we have CRISPR here. There are differences in the modes in which they're carried out, and of course differences in the timeliness that we can produce a desired change in the organism. So of the first ways that humans look to genetically modify their crops through breeding techniques was through obviously crossbreeding between individual crops. For years it went on and they would inbreed species based on desired genetic traits, but a relatively new idea in agriculture, or at least in breeding plants, is the idea of heterosis or hybrid vigor. And this is basically the case where two inbred parents of different varieties are bred to produce an F1 with more vigorous growth or more desired effects. So we can see here in the image with the cabbage here, P1 and P2 are two different parent inbred varieties, and the P1 P2 cross or F1 displays a more vigorous growth. You can also see it in the cross section of the cabbage head there as well. These pictures were taken on the same day of growth. Also in the image down here, we can see the same effect of hybrid vigor or heterosis. The middle is the F1 offspring, so you can see in the fully developed plant here, it's much larger than the two inbred parents. You also see the ear of corn displaying the effect, and then also in the germination phase, they're faster to take to growing. Now this seems like a relatively simple idea, but this actually wasn't really recognized in agriculture until the late 1910s, and the first hybrid corn species wasn't recognized nor distributed until around the 1920s. Corn production, they didn't use hybrid corn extensively until about the 1940s in the United States, so it's a relatively new idea in agriculture. This slide we take a look at mutation breeding, and this is a method of genetic modification that was first practiced in the 1940s, and it involved using a number of mutagens in order to produce mutations in the genome of the plant they were trying to modify. So some of the ways in which they induce such mutations are with UV light. So UV light would degrade DNA, and in particular it was noticed that it would cause interstrand pyrimidine dimerization, meaning the pyrimidine base pairs would bond with one another and disrupt transcription. Another way was with radiation. They would use X-rays or gamma rays to also denature DNA. And lastly, they use a number of chemical mutagens. Ethyl methane sulfonate, or EMS, is a carcinogenic chemical that is believed to induce mutations from guanine cytosine pairings to adenine thymine pairings, causing mutations in the DNA in plants. What you see in the image here at the right is a pair of morning glory plants. The one on the right, the deeper blue color, is actually the wild type or natural form of the plant, and the one on the left has actually been treated with gamma radiation, which ultimately led to a lighter color in the flowers. Mutation breeding commonly produces effects such as dwarfing, but certain effects can be selected for, because in addition to dwarfing, it could mean more flowering or a heavier fruit set, things of that nature. It's important to keep in mind as well that with mutation breeding, although it is rather effective at producing mutations, the mutations that you get and the effects are entirely random. In this slide, we take a look at agrobacteria-mediated gene transfer. And this is one of the first ways that geneticists were able to input foreign DNA into the genome of the plant that they were trying to modify. So in this case, they noticed that Agrobacterium was responsible for the formation of what's called a crown gall, or this tumorous growth here at the root crown of the plant. And through further study, they noticed that the bacteria were using a plasmid to transfer DNA into the DNA of the plant cells, and this allowed for the tumorous growth, which harbored the bacteria, as well as produced open, which is a energy source that was specific to the bacteria. So in this slide, we see an image of the agrobacteria-mediated transformation, and these are the steps that geneticists would go through in order to induce new DNA into the plant genome. So obviously here the plasmid is removed from the bacterium. It's named the TI plasmid, and that just designates that it's specific to the agrobacterium. They would cut it with a restriction enzyme, then design an DNA fragment, or derive a DNA fragment from a different organism, insert the gene into the plasmid, then under certain conditions they would reinsert or allow the bacteria to take up the plasmid, and then under further biological conditions they would see that the bacteria would infect the plant cell and transfer the DNA into the cell's chromosome, where it would then be grown out and the desired effects would be seen. Now, as you can tell, there's a lot of steps here. The fact of designing a DNA fragment, removing, then reintroducing the plasmid to the bacteria is going to be rather difficult, and then as well as reproducing the biological conditions in which this bacteria species will attack plant cells is nearly not impossible, but fairly difficult. I introduced an example of agrobacteria-mediated transformation, and this is the technique that was used in the production of what was called the flavor saver tomato. And this is actually the first genetically modified plant that was FDA approved in 1994 to be mass produced and distributed. It met the policy that was stated in 1992 that GMO plants must meet the same requirements, including the same safety standards as foods derived from traditionally bred plants. So I thought that quote was rather interesting. This is taken directly from their website, and it seems rather conservative to me. Basically, this new introduced technology was subject to the same testing that was done on normally bred plants. So this tomato was produced by a company called Calgene, which is now owned by the giant agrochemical company Monsanto, and it was produced in Davis, California. So in this slide we see the ways that the flavor saver tomato was bioengineered. They first were able to recognize the gene that produced the PG enzyme, or polygalactoneurase enzyme. The PG enzyme is the enzyme responsible for the hydrolysis or the breaking down of pectin, which is the stable form of the starch in the tomato. It gave the tomato its shape, and basically the longer you could maintain that starch structure, the more stable and resistant to rotting the tomato would be. So they did this by introducing a separate gene that produced antisense mRNA. So effectively it was a complementary copy to the mRNA, and when the two were formed together they would form this complex that would effectively block the ribosome from translating the mRNA to produce the PG enzyme. So then you wouldn't have breaking down of the tomato, and the tomato would have a much longer shelf life and stay more firm for transportation. This eliminated the need for the common practice at the time, and is still in use today, where tomatoes were picked unripe for transportation because they were firmer and they would have a longer shelf life to get to their destination before they were treated with ethylene to ripen them. So the flavor saver tomato was able to ripen on the vine naturally because even though it was ripe, it had a general resistance to breaking down before it could be brought to market. Briefly discuss CRISPR-Cas9, and CRISPR stands for the clustered regularly interspaced short palindromic repeats, and the Cas9 is a CRISPR associated protein 9, which is the two together form the complex of the enzyme endonuclease that's guided by an RNA sequence to remove viral DNA in bacterial genomes. It's effectively the bacteria's immunodefense system against introduced bacteriophage DNA. CRISPR-Cas9 can be used in genetics in the sense that guide RNA can be designed to guide the endonuclease to a specific location on DNA. This has become easier through the years with the introduction of whole genome sequencing, where we can now find genes to guide the protein to. Once the cut has been made, you can either delete a gene by allowing the cell to reform the DNA by non-homologous end joining, where two compatible ends will come together, or you can introduce a new section of DNA where the cell will use that section to repair the cut site. So it's extremely accurate. You can guide the endonuclease to make mutations at very specific sites, and this has been used for a number of modifications in agriculture. So here in this slide we see the GMO crops that are FDA approved, and according to the FDA's website, this is an exhaustive list of approved crops. It may not seem like a lot, but one thing to note is that these are used in a variety of ways, such as food fillers, thickeners, material production. Some of these are used in the production of adhesives, and it's also important to remember that although it's only one variety, and here I'm kind of focusing on corn because that's the United States cash crop. 92% of the corn that's planted in the United States is genetically modified. This is true for cotton as well. It's 94%, and soybeans are also 94% of the crops planted, according to a study in 2018 that I found on the FDA site. So here in this slide we see the example of waxy corn, and this is a genetic modification that's commonly selected for, particularly in a food and industrial setting, since that waxy corn produces almost 100% amylopectin, which is a better starch for food thickeners as well as in an industrial setting, such as in the production of paper or adhesives. This waxy corn has been produced through breeding methods with near perfect results. Basically breeding out the WX1 gene that produces an enzyme for the elongation of glucose polymers into amylose starches. Such starches are not necessarily preferred if you're trying to use them for food or industrial production. So it's important to note that this can be selected for through breeding methods, but CRISPR-Cas9 has been used much more effectively and efficiently to target these WX1 genes and knock them out in order to produce waxy corn for the specific purpose that they're looking for. I did notice through research is that the definition of a genetically modified organism does change depending on what industry you're a part of and what lens you're looking at the organism through. One definition carries that a bioengineered species, or GMO, must display a feature that could otherwise not occur naturally. So we definitely saw that with our bacterial transformation, where we specifically introduced a new gene to the genome of the organism we were trying to modify. But one thing that's interesting about CRISPR is that it's relatively efficient and very accurate, and also we can produce mutations by making very specific cut sites or removing very specific sections of DNA, and these are mutations that very well could occur naturally. So that may change the public's perception of a genetically modified organism, when using CRISPR. In addition, CRISPR, being that it's very efficient, could be used in the future to genetically modify plants that will be necessary for the changing world climate that we see and the higher demand that we see with the rise in human population. And then lastly, it's important to remember that GMO crops are used for a lot of different things rather than just what you're going to see in the grocery store. So I hope that you learned a little bit about CRISPR and some of the genetic modification methods that are currently in use today. And I hope you enjoyed it. Thanks for listening, and here are my works cited.